Hello? Hello?
Yes, sir. Namaskar.
Namaste, sir. Namaste.
Ekaj minute, sir. Ekaj minute. I will just. Am I audible to Dr.
Yes, sir.
Gaiko?
Yes, sir. Yes, sir.
Yes,
You
sir.
are audible.
Right, sir. Right. Allow me to share my presentation. I just need to check if my presentation is. Somebody else is presenting screen. I'm sharing screen. Could you please discontinue that? Right, sir. Are you able to see my screen now?
Yes, sir.
Yes, sir.
Right. Perfect. So I am ready. Do let me know when I should start. Thank you.
Yes, sir. Thank you. Deshmukh, sir.
Yes, sir. Yes, sir.
Can we
Good morning,
proceed?
sir. Borkar, sir. Namaskar.
Borkar, sir.
Yes, sir. Namaskar. Very good morning to everybody.
Very good morning. Very. Deshmukh, sir. Can we proceed?
Yes, sir. Yes, sir.
Okay. Thank you. First of all, good morning, everyone. I warmly welcome speaker, Dr. Manoj Borkar, convener and head of the department, Dr. Ashish Deshmukh, professors, researchers, teachers, my colleagues, research students, and other dignitaries who have joined for this lecture. Today, on the last day of the lecture series, Dr. Manoj Borkar is with us to give a lecture on technology-driven wildlife research. Dr. Manoj Borkar, sir, and I have been in touch for a long time, even though I find it a little difficult to introduce Dr. Manoj Borkar, sir, because this person is famous in the fraternity working in the field of biodiversity, wildlife, and man conflict to the animal or environment. One thing in particular I would like to mention is that sir always writes articles on biodiversity, animals, or their problems. In it, they express their views on the conservation of biodiversity and make the concerned department aware of their duty. Another thing I would like to mention here is that while reading his articles, there are some words that are new to me, and you have to open the dictionary to know the meaning of those words. Dr. Manoj Borkar, sir, is MSc and PhD from Mumbai University, PGDEE, New Delhi, FVRS, Fellow of Academic Sciences, FIAES, MASCI, MISZ. I don't know the long form of these degrees or diplomas or the awards. He's a scientist and scholar in wildlife biology with erudition, versatile academician, a barefoot ecologist with ground connect, a poet, thinker, committed ecologist, author, lexicographer, who is a logophile, columnist, first awardee as a Goa State Meritorious Teacher, former member of Goa State Biodiversity Board, State Wildlife Board, former member of Goa State Expert Appraisal Committee, founder of Biodiversity Research Cell, Department of Zoology, Carmel College, Goa, and now he is a head at the Department of Zoology, Carmel College, Goa. An acknowledgement of what I have just said about him is that he has been honored by the Goa State Biodiversity Board with the Goa State Biodiversity Conservation 2021 award. With this brief introduction, I request today's eminent speaker, Dr. Manoj Borkar sir, to start his lecture. Thank you all.
Namaskar, very good morning to everybody, and thank you, Dr. Sunil Gaikwad, who's of course my good friend and fraternity colleague. He's been very modest, but we have a lot of intellectual exchange, and currently we collaborate on some very interesting research programs. So once again, thank you so much, Dr. Gaikwad, for this opportunity. It's always a pleasure to be a part of any event organized by the Department of Zoology of this prestigious Shivaji University at Kolhapur. And thank you in particular for allowing me to dwell on this very interesting premise of technology-driven wildlife research. I am not very sure whether the electricity would, be with me today, but in event of electricity failure, please bear with me. I will try to reconnect at the earliest moment possible. So yes, I believe I have got about an hour or so. Ek taas hai na majagade. Gaikwad sir.
Sir, you can.
Wonderful, wonderful. The longer the better. But I will try to, consolidate all my thoughts and try to disseminate a little that I know about wildlife biology. At the first instance, let me be very clear. I don't claim to be an expert per se. I'm still a student of wildlife biology, and in our lifetime, there is so much to learn. I guess nobody can claim to be an expert in absolutely any domain of expertise, as a matter of fact. So in the last 34 years, with a little field exposure that I've had both within and outside the country, I've been fortunate enough to travel all through the length and breadth of the country, barring a few states probably. I guess Jammu and Kashmir Kashmir is the only state where I have not been able to make it, despite several invitations for, some kind of anxiety and apprehension about the safety and security of that place. But I believe if everything goes well, very soon we could also be doing some work in that part of the country. We've come a long way. In fact, I should say wildlife biology as a domain is at its crossroads today. I remember very clearly when I was a student of wildlife biology way back in the 80s, our professors would always emphasize an extremely important advice, and that was, if I may quote my professors then, they would say what it takes to be a good wildlife biologist are essentially only three things. A keen eye for observation, which of course very much holds good even till date. Patience and perseverance, very important. You need to have loads of patience. More often than not, when we kind of sit glued before our TV sets and get enthralled with the Discovery and National Geographic, episodes, we presume and we almost think that the minute you enter a forest, the entire wildlife of that area should, march and line up before you. That's not really how it works. And therefore, as I say, maybe your first instance or the first time that you enter any protected area or any wilderness, it's quite unlikely that you could be greeted by absolutely anything. It could be well barren, silent, nothing much to stimulate you visually, intellectually, nothing. But I guess that's where your patience and perseverance pays. Probably the second time, the third time, and maybe, a number of visits that follow could open up new vistas into your understanding of the reaches of that forest and give you a better insight into understanding wildlife and the various nuances that a layperson may not really be fortunate enough to witness. So yes, in the first place, as students of wildlife biology, and I emphasize the word students, we are all essentially students, maybe with a little expertise on certain, I would say, dimensions of the entire spectrum of wildlife. We have to be guided by a few principles. That's one, you need to be very observant. You need to be having loads of patience, and you need to not give up on your perseverance. And third important point is you have to be able, it's not enough to just gather data. You need to collect, you need to collate, and you need to analyze that data. And by analysis, I mean you need to come to some kind of a statistically confident inference that would be accepted by your peer professionals. I mean, you can't just make sweeping statements about something that you see as a matter of serendipity or as a matter of fluke, or it's probably something which does not regularly recur, and you end up with the impression that this is how it happens. That's going to be a flawed observation that may not be statistically confident, because a bias would have crept in your observation. And therefore, to infuse some kind of strength, some kind of confidence, you need to rely on multiple sets of observations. And I think that's very important. So let me start opening up the whole discussion by telling you that in 21st century, maybe in 2021, as students of wildlife biology, it's not enough only to be looking at the conventional methodology. Now you need to bring in a lot of technology. So the technology-driven wildlife research saves a lot of time. It infuses a lot of confidence. In a sense, you are able to produce and generate irrefutable evidence for peer scrutiny and for peer validation. So that's again very, very important. The third important point is we now need to be a little techno-savvy. I would not say that we would be able to sort of master absolutely everything that's on the platter, but a few things that are pertinent to your field of research is something that you certainly should be more familiar with. So that's the premise of my talk today. I mean, the images that you see on your screen probably are what are going to reflect in my discussions. You've got a bed there, probably the flying fox, the megachiropteran species, which has come into a lot of focus due to the present extraordinary pandemic times, as a zoonotic carrier a lot of viral load. Everybody is quite apprehensive and worried about the spillover interface. There have been in the recent past quite a few of the zoonotic pathogens that have spilled into human populations. Just to mention a few, of course, the SARS-CoV-2, then you've, of course, also got the Nipah virus and so on and so forth. And there's a great deal of modeling happening in some of the leading labs of the world, including some of the topmost virologists that I've had the opportunity of inviting to be part of my own webinars, have cautioned that it's not going to stop here. In fact, this is only the beginning. That we are going to have maybe series of waves of zoonotic diseases. And therefore, I think one important area for all of us as students of wildlife biology is to kind of be able to identify those species which are of zoonotic significance in our own areas. So that would demand of us to undertake some intensive biosurveillance, not only in terms of identifying the species, but also be able to understand the potential spillover interface, maybe do some kind of serosurveillance, or maybe do some sort of microcosm studies in terms of what kind of viruses and pathogens do these bats really harbor, and what is the likely interface where the virus could eventually get transferred to the human population. So this is quite interesting again. And then on the left, you've got this camouflage robotic camera, which many of you would have probably seen in this Nat Geo or Discovery serials, that minimizes the human wildlife interface. You don't need to get too close and proximate to wildlife, lest you remain safe. The animal also is now very and cautious. Remember, any animal, the minute it realizes that it's being watched, it will stop behaving normally. That's a thumb rule. So it's always safe to be observing animals remotely from a safe distance in your own interest of safety and survival. At the same time, trying to eliminate all those, I would say, extraneous influences and disturbances that could interfere with the animal's natural behavior. And that's where these kind of robotic camouflage camera, I'm going to talk about this in details a little later through my slides. And on the extreme right, you can see a male frog with inflated vocal sacs. That's to suggest that I'm also going to talk to you about aneuron bioacoustics, which is an emerging area which attempts to assign to each aneuron species a specific bioacoustic signature. So again, this is pretty interesting because now you don't need to actually carry out any kind of physical collection, no physical survey per se. And if you have the requisite equipment to record, and then of course, subsequently to analyze, you got a lot of softwares coming from Cornell Lab in the United States of America. And I think some versions are free. If you really need to get into more advanced analysis of the vocalization of these frogs, then you need to be trained at that. But even for an amateur person, you've got a lot of user-friendly softwares which you can install in your laptops and do that. So I mean, I'm just trying to explain to you that bioacoustics is an emerging, and that also applies to the bats, by the way. So I mean, that's how we are going to, or rather, I'm going to take you through the whole presentation. I hope this slide is able to change now. Well, yes. So this is what I thought I would first explain to you. Just a minute. I am not sure why I cannot. Just a minute, sir. Right. Thank you. Am I audible? Dr. Gaikwad, am I audible?
Yes, sir. Yes, sir. You are audible.
Right. Thank you. Thank you. I just want to confirm that. All right. So let's start with something which is very obvious and something which is, a passion of the present generation. Everybody you see going out to the field, equipped with state-of-the-art cameras and telephoto lenses, and you've got the social media platforms where you can, the minute you take an image, you can immediately post the image and you kind of become popular in your own social circles, and you receive a lot of thumbs up and a lot of wows and whatever, right? So I believe I need to speak to you on the importance of this kind of photo documentation, which nobody can deny is very important. Good quality photographs are extremely important. That's a basic visual evidence of the presence of any species in a given place. And therefore, if you want to talk to people about what needs to be conserved or what needs to be managed from a wildlife manager's perspective, you obviously ought to produce documentary evidences, or may I say, photo documentary evidences to push your point. I mean, you want to enthuse people, you want them to listen to you. Obviously, you need to put up an image which will make them feel good about it. And therefore, the importance of good photography is undeniable. It's very important. But I'm only trying to suggest that we should not stop at that. You've got some amazing macro photography that people are into. Like, for instance, the image that you get to see on your screen is a close-up of a dragonfly. It almost looks as if it's talking, or it's probably, laughing, a kind of thing like that. So dragonfly ordinarily isn't something that would enthuse a lot of people in terms of conservation efforts, because this is kind of the lesser known, or not a very charismatic species in a sense. If you compare that with some of those carnivores, which steal a lot of limelight and probably sap a lot of funds, I don't think there are any takers for funding any proposal with dragonfly research. It has to be there only out of your pure passion and a sense of commitment. But then photographs can do a lot of difference. You talk to people, you probably have some popular lecture series, you write articles, you try to, garner some sort of public support. It can go a long way. So yes, photographs have a major influence in creating awareness about what kind of wildlife exists in a given place, which species need to be prioritized, how do they look? I mean, there are many people who wouldn't even know. Just to share a small, incident from what happened yesterday. Yesterday, I wrote my column on chasing the cheetah. Some of you who have logged in probably would have read this. I mean, since I write a regular column in one of the popular Goan newspapers here, the thematic focus yesterday in my column was chasing the cheetah. The entire, I would say, historical on how it started and what is the contemporary status of the whole, program. Trust me, a lot of people who kind of called me up or send me messages said, oh, why the hue and cry about cheetah? There are so many of them in Goregaon, in Mumbai. I mean, mind you, these are not lay people. These are extremely qualified professionals who probably do not understand the difference between leopards and cheetahs. Trust me, I'm not exaggerating. This is factual. They said, look, there are plenty of leopards in Goregaon, RA colony, and there are plenty of leopards everywhere. In fact, probably every week we have a conflict report appearing in the paper, and I have to tell them that's all about leopards. So the confusion is, what is a cheetah, what is a tendua, and what is a, I mean, in Marathi we say bibra, barobar hai sir? We say biptia. Or in Konkani we say bipta, right? So sometimes, and prima facie, if you look at it, if your eyes are not trained, probably people get confused between a cheetah and a leopard because one has rosettes, the other has spots. But the characteristic teardrop mark, which you see on the face of a cheetah, is what sets it apart from the leopard. So, I mean, I'm just trying to drive in a point. I'm not trying to sort of blow up and exaggerate about the ignorance people have in understanding the difference between a cheetah and the leopard, but trust me, it is there. But quite a few people still confuse between the two species. But the minute you show them a photograph of a leopard and a cheetah, and I mean, the look of it itself sets the two apart. So I think good photography is very essential. And it's not just about documenting, species, but you document species in the habitat. So I think that serves a huge purpose. I mean, you have a look at this. Many of these hummingbirds, many of these sunbirds, if you look at the iridescence in their plumage, it's something very exciting, something which really, impresses you. You have a good resolution. You've got a good close-up that really, steals a lot of, I would say, attention. So yes, this is very important. May I just remind you, we're talking today about technology-driven wildlife biology. Trust me, until, let's say, 2 decades ago, we did not have mobile cameras to the extent that we use them now. So today, your mobile is, it's got a built-in camera, which is pretty good. I mean, and you will accept that gives you an advantage that you needn't actually be carrying those very cumbersome equipment to the field, in a bid to capture what you see. Your mobile is good enough. That's the charm of technology, and that's the benefit of technology if you use it properly. Not, of course, for taking selfies. I mean, that's again up to each one of us. And what exactly would you want to capture as an image, your own self or something which is more beautiful and more brilliant that you have out there, right? So that's something important. Then let's take a quick look at camera traps in wildlife research. As I said, I still remember it's not very easy to see wildlife, and certainly not when you want to see it. I mean, wildlife does not synchronize or sync with your free time and your leisure time to, as I say, parade before you, as in when once in a blue moon you visit a forest and you expect everything to line up before you and stand in attention, give you enough time with poses for a good image is not possible. And therefore, this is something that you all need to understand, that if you want to capture images even at a time that you are not able to be in the field, I think next best option is, of course, a camera trap. And this is an extremely precious, non-invasive data collection device, which can, I believe, throw a lot of light, not only on the kind of species that are present in your area, but also on their activity budget, their behavior, particularly crepuscular species, which don't come out in the open during the day. Probably you find them very active at dusk or dawn, and that's when you're fast asleep. Or you wouldn't probably want to, kind of take the trouble to go to the field or a field which is rather difficult to access because it's a very rough terrain. I mean, I still remember this statement which my teacher of wildlife biology would always tell us, that no matter how advanced your technology is and your protocols of field study are, the field shall continue to remain as rough as ever. It's not going to improve because it's wilderness. And wilderness is meant to be rustic. It's meant to be difficult. It's not easily accessible. And probably that's one reason the animals are there and not in the ease of your urban environments. I'm not trying to deny the fact that you've got a great deal of wildlife in the urban scapes too. But I'm talking about documenting wildlife in the most difficult terrains will require of you to certainly rely on camera trapping, because that can provide you a lot of information on its ecology, its ethology, its conservation future. In terms of, like, say, for instance, if you know by your camera trapping that there is, I would say, let's say a tigress with cubs, that's a good sign. I mean, that's an evidence it's recruiting to the population, right? It's recruiting to the population. Or for instance, if you look at this top right image, you see a hyena carrying a livestock. So you know that there is a conflict issue, that there is a depredation, there's a predation happening. And this could probably go against the conservation mandate, because if people are not with you and if they suffer losses, then they tend to be hostile. And the hostility doesn't take a long time to translate into anti-conservation, anti-wildlife sentiments. If they pick up, then not even the forest department can do much. As I always say, and I've been very emphatic, and I underpin the fact that if you want to be successful in any of your conservation mandates, you need to take the people along. And the only way to take people along is to minimize any kind of losses that they suffer or are likely to suffer on account of wildlife, be it loss of life or loss of property, loss of livestock, or some sort of a threat perception. That's certainly not in your favor, not in the favor of the wildlife either. So this is very interesting. So yes, a good number of camera traps, some of which are, I mean, they run on a battery. I think they can last for quite some time. You need not visit them regularly. You can probably visit them at some regular intervals just to retrieve the data. So this is a widely employed tool in wildlife research, not just research, but also monitoring in management. It can help you in throwing some light on animal abundance, animal movements. That's very important. I'm going to, I'm sorry. Is somebody marking my presentation? Hello?
No, sir. No, sir.
I'm extremely sorry. I think.
No, sir. Someone, Dr. CG, like that I have seen. Dr. CG, I could not see the surname, but one of the participants, Dr.
Correct.
CG. Yeah, please erase
Right.
it.
That's what
Red
I
line
noticed.
is there.
Somebody is overwriting on my presentation.
Yeah, red line I can see.
Unless the person has something to question, I would be more than happy to respond to that. But please do not overwrite on my presentation. I mean, not for anything. It just distracts me, please. And I think it's not readable. Can I just wait for the person to clear
Yes,
that
sir.
off?
We will find it. Dr. CG, yeah, it's only Dr. CG. There is no surname. Dr. CG, we request you to erase it.
I mean, if he does not, if the person is not agreeable with what.
No, actually, how he can draw something, because.
No, unless it, I hope it has not happened from my side. I don't think so.
No, I have seen. When that line was appearing, the name was appearing there.
Yeah.
All right, fine. All right. If the person may not know how to do away with it, he has to just undo it. That's it. All right, doesn't matter. I think we'll take it further. Doesn't matter really, because it's not unless it's going to kind of.
Sir, it will persist in all the slides.
That's I'm sorry, I'm not able to do anything about it. If the host is not able to do anything, I don't think even I would be able to do anything about that.
Dr. CG, are you there?
It's all right, Dr. Anna. It's all right. I think he may not be able to, he doesn't know how to undo it.
No, I will tell him, sir.
All right, may I continue in that case?
Yeah.
So what I was trying to explain to you is you got good number of camera traps available today, which is not very expensive, and they are being extensively used by most forest departments, as also by research students to understand critical information on many dimensions of wildlife, right? So I think this is something very important. One very interesting spin-off of camera trapping, if the camera is installed in, I would say, in a strategic location where it cannot be tampered with, is it can also catch images of, as I put it, some kind of potentially serious, innocuous, or serious criminal, tendencies. For instance, if people are setting a trap, normally they set a trap on the regular path of animal movement. So I think that could also be caught in the camera, and some sort of preemptive action or some sort of a mitigatory action can certainly be taken. So this is very important, that camera traps or trail cameras, there are motion sensitive, there are sensors in infrared sensors. The minute the animal moves, it kind of trips and you have a flash which captures the image. So this is pretty interesting. I'm not getting into the technicality or the nitty-gritty of how exactly it works. That's not the purpose. As much as the purpose is to tell you and make you familiar with the different gadgets or the different techno innovations that are available to us today. So this is again very important. They come in wide variety. You've got a small collage here to see. And something very interesting is animals, once they note or spot this kind of installations on the barks of trees, they also become pretty curious. They come, sometimes you get very interesting images like this, where you see the leopard, kind of coming very close to the camera. So you get very interesting and sometimes very comic images as well. But this is very important. I need to tell you, insofar as the state of Goa is concerned, I think the presence of tigers in some of our protected areas was confirmed. Evident from the camera trappings. I think it's not just about the camera trappings that indicate the presence, but also it helps you, for instance, in certain carnivores like the tigers, which have got stripes specific to individuals. It can also help you in understanding the conflict potential of a given individual. For instance, if you want to identify a rogue animal, which is probably regularly cattle lifting, or it's probably threatening people in the adjacent human settlements, by studying the stripe pattern, which probably has been captured in the camera trap, you can compare that with your referral database and know exactly with great precision, without any iota of ambiguity, that this is the animal which is actually creating a problem. So that's, and I need not say, with COVID-19 restrictions, many of us have not been able to go to the field. And that's when I think the next best option, of course, is the camera trapping. I think it offers you an opportunity to carry out uninterrupted fieldwork, which would have been rather very difficult otherwise. And of course, as I said in the beginning, your regular presence in a given place could compel the animal, because they don't just rely on visual cues and clues, but they've got very sharp olfaction. So I think sometimes it's good not to frequent a given place on a regular basis and maybe exercise any of these options for remote study. So I'm just going to change my slide here. Oh, I'm really feeling sad that the mark is interfering with the whole screen. And that's how it's going to be recorded. So this is really a bit, I don't know.
सर परत तुम्ही ते क्लोज करून चालू केलं तर निघून जाईल असं मला वाटतंय.
Shall I?
If you close your presentation and again restart,
I will do that. I will do that. I will do that. I will try to do that, please. Let me see. Just a minute, sir. All right. Let me check.
yes,
Is it being shared,
Yes, sir, it's gone.
yeah, it's gone.
Thank you. Thank you. Thank you. So, I mean, was it from my side in that case?
No, sir. No, sir.
Thank you. All right. So I think this is going to be, again, pretty interesting for you, taking a whole discussion on remote technology to capture images. This is something extremely fascinating. And you may wonder, since we're all zoologists, one animal or one insect that we're all overtly familiar with, of course, is the cockroach, which is a household pest globally, barring probably the polar regions. But I'm just going to talk to you about that in 2 minutes from now. On the left, you see this kind of robotic cameras, which are mounted on wheels and camouflage to look maybe like some sort of an object that could look similar to maybe a turtle or a terrapin or something of that sort, tortoise. And you've got the remote control whereby you can maneuver the camera, which can be, as close as you desire it to be, subject to the animal not getting too curious about it. Sometimes you often have these instances where the carnivores start biting it or chomping into it and destroy it also. I mean, there are a few cases on record where lions have simply got angry with this sort of an intrusion. Something which comes too close to it is something which irritates some cats, and they could probably sort of destroy. That's a possibility. Then again, on the lower slide, you've got the thermal sensors, which are very useful in conducting nocturnal observations. Certainly you can't be using your flashlights and stuff, and you would have seen this in some of those extraordinarily well documented films in National Geographic or Discovery Channel serials, where if they want to study how exactly does a predator prowl and eventually ambushes the prey, and the kind of panic it sets in the entire herd of the prey, I think the best way to do that is not by switching on any light, because for conventional photography, you need light. And here is an option where you need not turn on the lights, but you just simply have a camera which is very sensitive to, I mean, that's a thermal imaging camera, the heat radiated by the body. These are all homeotherms, by the way. And even if it's a poikilotherm, there's a certain amount of metabolic heat which is generated that can be sensed by the camera. And that's how, and it's a real-time camera again, so you can catch movements. That's again fascinating. But what I want you to particularly focus on is, not many of you may be familiar with how micro robotics, I think on day one, when Dr. Kamath spoke, I remember very vaguely, he did speak about some robotics. And he said how certain things have influenced, robotic designs, if I remember it correctly. To take that discussion further, what you see on your screen next to the cockroach is a micro robo inspired by the cockroach itself. And it's so small and miniaturized and compact and mobile that it can simply creep into any kind of a crevice. I've given particularly the dimensions of this whole micro robo, which is hardly around, not even, I would say about 0.9 inch, which is very small. Its total weight is around 320 milligrams, right? Its speed is astonishing, which is about 13.9 the body length in, which is about a foot per second. If you compare that with the cheetah, incidentally, which is the fastest mammal, right? It's a little less than that of a cheetah. I'm just talking comparative terms. So if you look at all this, I mean, even the gait is pretty good. It's able to kind of move, bounce, jump, and trot and trunk and all these kinds of maneuvering. Ergonomically extremely pliable, and very important, it can carry a payload which is 10 times its own weight, which means around 320 into 10 times, which is fascinating. I mean, if you may ask me, how does it help really? If there's some crepuscular species, which is furthermore fossorial, which means it's burrowing, you certainly would not be able to insert any kind of a camera, inside burrows. The next best option you have is something that has been devised by, incidentally, an American or Indian origin engineer, Jairam at Harvard University, and he calls it's Micro Robotics Department of Mechanical Engineering, and he calls it Hammer Junior. That's the name given to this robo inspired by the cockroach. So those of you who thought, well, we've been dissecting cockroaches as and when that was allowed, and how does actually that translate into any kind of application, where here is an application that a cockroach is a model organism for micro robotic inventions. It's quite fascinating, and I think we should all be glad about that. I'm just going to change my slide further. I hope I'm just going the right sequence. Just a minute later. I'm just confirming it, please. Yes, I'm so sorry. Apologies for this interruption. I'm sorry again. Right. I'm just confirming. Is my screen visible and am I audible to everybody? I just need somebody to confirm that, please. Sir, am I audible? Anybody, please? I presume I'm audible, and I take you further into understanding how the geographic information system can, I would say, add synergy to the efforts of the wildlife biologists. In the sense, if somebody thought that geographers have nothing to do with wildlife biology, that's a fallacy. And geographers, in fact, can do a lot of handholding today in present times. In any study of wildlife, the spatial dimensions are really very important, because you need to relate everything that happens in an ecosystem to the space. The dimension of space is critical. It's all got to do with how much of volume, how much of space that a given event is actually happening, unfolding. Any kind of observation that you seek to unravel the mystery and understand the nuances of needs to be viewed in the light of space. So for understanding space, you need a geographic information system, which is a software. Now you've got multiple layerings. In fact, I borrowed this from the Wildlife Trust of India, the slide on the left, of course, on how exactly is a GIS consolidate and organize this specific information on a spatial platform. And by spatial platform, I mean space. So you've got multiple layers, as you can see here. I'm so sorry. You can see multiple layers here. For instance, I'm just not able to see them very clearly. Right. So if you have, say, in a given ecosystem, you've got multiple attributes. You've got a landscape or a waterscape, which is the lowest layer here. Then you've got man-made attributes like roads and bridges and all kinds of constructions. That would be the layer of road and the railways. And then you've got this human activity layer. For instance, you could be having agricultural plots, you could be having pastoral lands, and you could be having some sort of an activity happening. So that will be the human layer. And last but not the least, in a given space, you also have a layer which is constituted by your animals of interest. That's a specific interest to you. How do you integrate these multiple layers to build a consolidated impression, which is very holistic, which puts the animal of interest at the center stage, but gives you a linkage to all the attributes? And that is the beauty of GIS. And I've got two specific examples to share with you. Before that, of course, I just want to tell you that all types of geographical data, not only in terms of, I would say, the spatial context, the location of the place, because you also get the GPS, geographic positioning system, you get specific geographic coordinates. For instance, if you say that you did cite an animal which is critically endangered, obviously the next question that would be thrown at you is, what are the specific coordinates and where exactly do you expect these conservation initiatives or interventions to happen? And if that is the question, you need to be able to furnish the exact space or spatial context, which is possible with GIS. That's what I'm trying to tell you. So in fact, the recent efforts, even in Goa, because I've been associated with a couple of wildlife management plans for two protected areas of the state. So we have also used this GIS platform to consolidate information that's critical to conservation management. So what does it tell you? It tells you the specific location, it tells you the quantities, it tells you the densities, it tells you what is happening within that area of interest in terms of anthropogenic pressures. You're also able to monitor the change in time. I mean, if you want to go by a retrospective analysis and comparison, in last decade or 10 years, how has this particular landscape changed? Whether it's degrading, whether it's improving, whether it's static. These attributes one can easily find out from the use of GIS in wildlife biology. In fact, today, no study on wildlife biology is complete without an element of GIS being incorporated in your database. I've got two examples to state. These are the corridor use of the bear. And the scale that you see here, you can see the scale on the left. This is basically the conflict in terms of the difficulty that the bear experiences in moving from one habitat to the other through the corridors. It encounters a lot of problems. So what exactly are these problems and what is the intensity of these problems is something that has been plotted to create this GIS database on the difficulties that bears experience and encounter as they move between habitats through the corridors. So you've got a scale which is superimposed on this entire geography of, that's the area of interest. Again, if I may quickly talk to you about this, this is a part of the Northeast where they are talking about the habitat characteristics of the Hulex gibbon. And I'm sure many of you here would be in the knowing that the Hulex gibbon is an important species because that's the only ape species that we have in this country. The only ape that we have in India is the Hulex gibbon, which you find confined to the north. It's more endemic to the northeast. So obviously, from the perspective of conservation management, or monitoring the population, particularly in terms of the changes that are rapidly noticed in the landscape or the habitat. You want to monitor the habitat. You want to bring about habitat improvement. You want to leave certain regions inviolate for any kind of development, as you put them, no development zone. You certainly would have to chart the various uses that that entire landscape is put to. So this that you see on your screen, I'm not sure if my cursor is visible to you, but this is again a GIS a tool that has been used to evolve a map on land use patterns and the habitat characteristics of the Hula-Gibbon landscape in Northeast. So just to tell you how important is GIS. And if anybody is here in this webinar right now listening to me from one of the similar events hosted by a college in Goa where, Smith, please. Bilal Habib from the Wildlife Institute, who's also a carnivore biologist, he made some presentation, a very good presentation where he spoke about the carnivore movement ecology. This is something new. I think we are still trapped in, EP, ODM and ODM ecology, which is a classical ecology. I'm not seeking to kind of belittle that. That's the basis. But now in 2021, we are into, it's an era of specialization. So as you have, we no longer talk about general ecology per se. Now we've got specialized domains like movement ecology. What exactly is that movement ecology? A movement ecology has all got to do with the relationship of animals with the space and the movement there within. So, I mean, it's one of the common challenge in conservation management is to be able to keep a track and understand why and how and where do the animals go at different points of time. So if you kind of understand the temporal context of movement, animal movements, which is that the animals move in relation not only to time, but also space. And why do they do that? And is climate change driving this kind of movements, and how do they adapt to the new places? Is the resource potential of the habitat the trigger for the movement? Is it a kind of an endocrine milieu that has changed in the body of the carnivore with titers of certain hormones shooting up? Is that what is making the animal move? So there are a number of questions that you ask, but even for understanding this critical dimension of animal movements, you can always use a GIS platform. And this is basically what I have picked from Bilal Habib's recent paper. It's one of the recent paper in 2020 in, I think, Ecology and Evolution, one of the finest journals. Bilal is, of course, an exceptionally good wildlife biologist with the Wildlife Institute of India. But what I'm trying to explain to you here is, again, an animal, a wild animal, which is choosing to move. Why would it move if everything that it seeks is available in the immediate environment? All that it seeks is, of course, a space to colonize. It needs adequate supply of nutrition. That's a prey base if it's a carnivore. It needs water. It needs adequate opportunities to mate and pass on its genetic matter. It has to take care from competitors and predators. And if these things kind of become difficult, then the animal left with no option but to move. And movement, you know, is what permits and allows you to explore and exploit the environment very sustainably so that you don't overexploit certain critical resource. So movement ecology is very important. And I think GIS certainly is opening new vistas of understanding this movement ecology. That's basically what I'm trying to explain to you. Let's quickly move forward. And I need to tell you that this is some of the exemplary wildlife that we have in Western Ghats. You've got something like the king cobra, you've got the Malabar gliding frog, which is an endemic species. You've got a number of new anuran species being reported from the Western Ghats. It's a hotbed of anuran diversity. Varadgiri and the likes regularly around. Many of the herpetologists have been reporting new species from different parts of this Western Ghats series. Then it's also an important bird area. I mean, we have quite a few in the political jurisdiction of our state. And again, you've got a lot of reptiles, residual within the precincts of our, well, I am just now going to tell you, I mean, this is again very interesting. While all this time we associated the microscope with the biology practicals or zoology practicals or maybe microbiology practicals, I need to say this with a lot of conviction because we are doing this work at the moment. advanced microscopy like the scanning electron microscopy is a very, very valuable tool in understanding certain interesting and hitherto unknown dimensions of wildlife. And by wildlife, let me remind you, are not only the charismatic species, but anything and absolutely everything that exists in wilderness, except our own species and the species that have been domesticated. Sometimes you have cattle movements in wildlife, so nobody should argue that the cattle in wilderness is also wild. They could be at the most feral cattle and not surely wild. So here is a species on top left, which I've been studying for more than 20 years now. It's a whip spider. The first report from the state of Goa also was done by me and my students way back in 2006. And, there's a new emerging branch of biology called Bioprospecting. And it was reported recently that for some strange reasons, this particular arachnid, it's a very, I would say it's an enigmatic, bizarre looking, it's kind of scary looking arachnid, which is really not a spider. It's an amblypyge. It's a different order altogether. It's almost a living fossil, hasn't changed since last several, I don't know, millions of years since its evolution. And we have it you have it in your state of Maharashtra. I think Dr. Deshbandhu Bastwade, a very well-known zoologist from Western Regional Station, the ZSI, was the first one to report it from Sanjay Gandhi National Park. And I'm sure it's there everywhere. And from Goa, of course, we have one species called Phylogeny stripsoni. If you look at what you see on the right, the adjacent image, and I trust me, I'm sure you would not be believing that these are nothing but spherules that you see when you look at the surface integument. I mean, if you look at the turgle plates in this arachnid, if you observe them through the electron microscope, you find something that forms a sort of a layer on the surface, which seratogument, this is for seratogument, and this is what imparts a super hydrophobic property to the exoskeleton of this arachnid. Now, mind you, here is a potential bioprospective molecule, a substance which may find a lot of application in industry. So I'm just trying to explain that to you. This is a new dimension of wildlife research, that in many of the species that we study, we have not been able to bioprospect. Today, everybody will ask you, well, you want to preserve this animal. Why? How do you justify it? Is it just the ethical value that it deserves to live, that therefore you conserve it? Or is there some sort of a scientific value or some kind of bionomic value? And the answer is yes. Probably if by whatever techniques you are able to characterize that molecule, which I'm showing you here, appears like spherical globules on the surface of the integument, I think you would probably be able to unlock that secret of what is it that makes the integument of whip spiders super hydrophobic, which means water resistant, not just water repellent, water resistant. So this is again very, very important. I'm so sorry again. I think you have something very interesting here. I've got two figures adjacent to one another. Here is an insect which is probably feeding, and you can see very clearly it's picked up a lot of pollens. Mind you, this is very interesting. Palynologists who specialize in study of pollen grains have been increasingly using scanning electron microscopy to resolve this kind of pollen grain morphology, the microsculpture. What exactly facilitates these pollens to stick to the surface of the body of the pollinators, right? Entomopollinators. These are the insect agents that pollinate. So again, here is the technology. Scanning electron microscopy is something which allows you to get a three-dimensional visualization of structures, the topology, the microsculpture, and I think that's incredible. There's something very, very interesting. I'm also going to speak to you quickly about wildlife forensics, which I believe is an indispensable tool. And the image that I've used here is something that we can't probably imagine in our country because it's prohibitively expensive, because it's an ultra modern state-of-the-art forensic lab for wildlife crime. And I think this is a combination of multiple domains, from molecular biology to chemistry to geology. It's kind of amalgamation of so many domains which allow you to zero in on the exact cause of death of animals in the field, or maybe a victim of a wildlife crime. And then if you are able to compile and I would say come up with a dossier, a sort of a dossier or evidence which will stand the test of a judicial scrutiny in the court of law, then you will be able to prosecute the criminals and not otherwise. More often than not, people escape from this kind of enforced prosecution because they escape for want of evidence. And unfortunately, in India, most of the wildlife criminals have been scot-free. Most of them, I say, not all necessarily, only because we do not have the requisite backup for wildlife forensics. I think this is very important, that the emergence of wildlife forensics is a boon. It's a kind of, I don't know, artificial intelligence which allows you to resolve the hitherto pending mysteries on the cause of wildlife crimes. I mean, it's got a lot of things. I'm going to, as I speak, I'm going to take you through many of these dimensions which could also have a consequence on wildlife forensics. So what exactly do they do? They have got experts from diverse fields who will help in piecing this story together and coming up with a more reliable and a more acceptable reasoning towards what possibly is the cause of death of a given animal that you see has died under mysterious circumstances. You just want to confirm this. So this is again very important. It also helps you in resolving taxonomic disputes, polymorphs, which may look different but could be the same species. That's also, and therefore there could be a kind of a jurisprudential issue where you are treating the same species which looks different as two different species. I think molecular forensics, I'm going to talk about which genes exactly are used in that kind of forensic approach. So this is again very important. It's fast, it's accurate, and it's very reliable for any kind of a criminal investigation where you need to produce evidence to the court of law for prosecuting a criminal who's been in your custody. I think this is very important because it helps you in resolving taxonomic disputes. It helps you in resolving spatiotemporal genetic divergence. I'll just give you a quick example. For instance, if you know the whole story of the reintroduction of cheetah from Namibia to India, the Kuno area, which has been designated now for the pilot trials of reintroduction of cheetah, one argument that was put up by some of our experts like Dr. Ravi Chilla, who happens to be a good friend, a very reputed carnivore biologist, is how would an exotic or non-native species, which is what the African cheetah is, at all survive in the Indian conditions? Mind you, let me remind you that what we had in India was, of course, the Asian cheetah, of which now the last, and that's shocking, 12 individuals, of which 9 are males and 3 are females, exist only in a small area of Iran. Iran is an Islamic republic that does not treat its conservation biologists with a lot of respect. I'm sorry to say that. In fact, very recently, 12 of the conservation biologists have been arrested and prosecuted and tortured on this suspicion of espionage, that they've been spying for Israel and the United States of America. And what was their crime? They only went to the field to collect the scat or the fecal matter of the cheetahs for a study, to harvest the DNA, to maybe do some sort of investigation. Unfortunately for them, the scat collection was done in the proximity of a critical defense installation. All the 12 guys were taken into custody. One has already died under mysterious circumstances. So that's the story about Iran cheetah program. Now, given this situation, you certainly can't be asking Iran for any cheetah, and they're not going to do that, because in the past, in the 70s, they'd agreed only on the condition that we exchange the Asiatic lions with them. That was Mrs. Gandhi's time. That's history. So today we are left with only one option, and that's to bring the African cheetah and see if it can adapt. One argument put forth by a lot of, I would say, skeptics is, if it's an African cheetah, how will it survive? And is it ethical and scientific to introduce a non-native species which tantamounts to encouraging an invasion by a non-native species. I hope you're getting my argument. But to that, this is very important. The wildlife forensic approach taken by the Center for Cell and Molecular Biology, there's a big number of workers. I mean, earlier we would speak about Lalji Singh. He is no more. There is a paper, I think very recent paper in 2020 again by Rai and others. I think around 8 of them from Africa, from University of Cambridge, from some of the best facilities in the United States of America, and of course the Center for Cell and Molecular Biology, where they have compared the mitochondrial DNA, now listen to that very carefully, of the African cheetah with a cryopreserved sample of the Asiatic cheetah, and they said it's matching, which means inference is very clear that the lineage of Asiatic cheetah that we lost in India in the 60s definitely was from the same stock in Africa. That's the interesting, I would say, inference from this study. And that's basically what I mean. This is what I mean by resolving taxonomic disputes. So now if you come up with this argument that if you compare the mitochondrial DNA of the African and there's no Indian cheetah, Asian cheetah, which we lost in India, it clearly implies that they have been imported from somewhere. They've come in from somewhere. And it's quite true that if you peruse historical records, somewhere in the beginning of the Africa to India, and that's probably the connect. So this is basically what I'm trying to explain to you. Genetic diversions, evolutionary history, origins, endemism, a lot of things. Sir, am I audible? Am I audible? Please confirm, am I audible?
Yes, sir. Yes, sir. You are audible.
Right, right. So the minute I'm off the internet connectivity, please call me on my mobile.
Yes, sir. Yes, sir.
Right. So taking the whole discussion of wildlife forensic further, I'm going to share a few of my experiences with you. Between 2012 and 14, I had this God-sent opportunity to work in the peninsular Saurashtra, especially the coastal region, looking at some interesting dimensions of human-wildlife conflict. And obviously, one conflict animal was India's best known and the most publicized and I would say the most flamboyant of the charismatic carnivore, the Asiatic lion. Once again, there was a lot of hue and cry that the lion that is spilled over from the metapopulation habitat is now causing huge conflict by lifting cattle from the local communities. The Ahir is one community. You normally talk about the Maldharis and the Rabbaris from the metapopulation habitat in the Gir at Sasan. But if you look at the coastal Saurashtra stretch, there are these pastoral communities called the Ahirs who regard themselves, they are basically Yadavs, and they consider themselves to be in the lineage of Lord Krishna. So for them, cattle is extremely, I would say, it's not just a matter of livestock, but it's something holy. So loss of a single cow or a milch cattle is something that infuriates them. And therefore, there have been a few studies conducted by Wildlife Institute of India. I remember very clearly. Those were the days that of course, we were working with the Gujarat State Forest Department. Some of these things were in, I remember Mr. Richard D'Souza was the additional principal chief conservator of forest, a man who extremely erudite and very supportive of the academia. He spoke to his counterpart in Gujarat and gave me that permission to do my 2-year-long study on human-wildlife conflicts and the community response. And I need to tell you, by analyzing the scat of the Asiatic lions, we were able to establish that the cattle, the scat is, in other words, nothing but the fecal matter, and a lot of non-digestible fractions from the diet, which of course is going to be a herbivore, bones and hair, which do not easily get digested by the digestive secretion. So they remain in the scat. And if you are able to sort of screen the scat, there is a procedure for that, and dry it and then try to assign it to that specific species from where it has originated. I mean, there are certain bones that an osteologist, an expert in the study of bones, would be able to say with a lot of conviction, well, this bone is definitely from this particular animal. So there are studies like this. The slide in between is a collection of bones collected from carnivorous cats, which means, you know, if you sort of look at it quantitatively, look at the number of times certain element of that bone appears in the scat, it tells you more or less what is the prey preference of that carnivore. So prey preferences can also be understood by scat analysis. And that's not going to be easy because it's a technique. You need a lot of technique. Similarly, what you see on the right is hair. It's a whole new area called Trichotaxonomy. And by Trichotaxonomy, I'm not sure if you got the image. I'm going to show you that later. A Trichotaxonomy is a very interesting applied area where you use hair morphology and the pattern or distribution of pigment in the medulla of the hair shaft can tell you whose hair it is, and therefore it's a very reliable indirect species specific evidence that you are able to collect from the scat to ascertain which animals constitute the prey base of that carnivore. So that's again very, very interesting. So people who specialize in study of hair, you will be astonished to know there is Indian Hair Society. It's a professional body of trichologists. So trichology is a scientific study of hair. And in wildlife forensic, these people play a very important role. So
sir, your voice is breaking.
So voice is cracking. Is my voice cracking, sir? Hello? Is my voice cracking? I want a response, please. Can you hear me?
No, sir. It's clear.
Right. I think some connectivity issue at that end. I'm so sorry about that. So I think wildlife forensic also is the main domain of expertise based on which, as I've said before, you can resolve a lot of this illegal trade in wildlife artifacts and trophies and whatever. So wildlife forensic investigation normally revolves very broadly in two ways. One is you rely on physical evidences, which could again mean you do necroscopy, which is again investigation of the dead, or maybe you investigation of the scat and stuff like that. You do microscopy, as I've just explained to you, trying to ascertain species specific evidences like hair or bones or shells, scales, ivory, claws, whatever. Or you probably rely on the imprints as the animal walks on soft substratum, which could be whatever, the mud or the clay or whatever. And there's a small image that I put here. And that's the pug mark imprint of a carnivore. And that's of a tiger, basically. So if you look at these imprints and understand the morphometry and the morphology of these imprints, not only would you be able to delineate and differentiate between the type of carnivore, but you also can approximate the age, the gender, the sex, and you can more or less build some information on the ergonomics, the gait pattern, and so on and so forth, right? So that's again very interesting. Whereas the wildlife forensic now also talks about integrating molecular biology, techniques of biotechnology and molecular biology. I just spoken to you some time back about the mitochondrial DNA, which are used with certain genes. I'm going to speak to you about at least these 3 genes. For instance, you've got 12S rRNA, there's one called 16S rRNA, and there's one which is known as cytochrome B or Cyt B, CYTB. These are what we call as forensically informative nucleotide sequencing. It's abbreviated as FINS, very easy to remember, FINS. Fishes have fins. So forensically informative nucleotide sequencing is a state-of-the-art approach in wildlife forensic that uses domain expertise from molecular biology. How exactly do you do that? I'm just going to speak to you about this a little later when I speak to you about eDNA or environmental DNA, which is again a very fascinating novel approach, which is technology driven, maybe molecular biology or biotechnology driven approach towards resolving wildlife crime. So this is again, I have to also tell you there is this very interesting software called mStripes, which is being used in India. For a long time, we relied on plaster of Paris casts and the markings on the glass pane of the the paw imprints, and try to calculate and understand the allometry of different parts of the paw, but that was found to be a little erroneous. And today we have a huge database which is collated in terms of the software called M-Stripes, which I think is being used across the country in all the forest departments, where you feed in that data and you are able to, that's a referral database which is stored in a kind of central place, and you by feeding in your data specific to your field and markings and whatever measurements, you can more or less ascertain whether it's a particular animal which has already been documented before, what exactly is the direction in which it is moving, if there are any new individuals that have come into this territory. A lot of these things can be made out. So that's again pretty fascinating. I'm just trying to remind you, we're talking about technology-driven wildlife research. We have strayed far away from the conventional approach of go to the field, spend hours together in a hide, and you carry with your binoculars and a notebook, and that's what we were taught, that you need to spend hours and hours and hours in the field observing, making observation. I am not saying that this is no longer valid and that it's outdated. I'm only trying to tell you that technology complements it, and technology takes it to the next level. So this is about molecular phylogeny. I just explained that to you. Molecular phylogeny is nothing but using amplifying DNA fragments and trying to compare that with referral database using certain genes. I mean, invariably, this is done from genomic DNA, I mean to say from nucleus, or it could be also done from mitochondrial DNA. I think mitochondrial DNA harvest is more easier. It's kind of circular DNA, sort of a strand instead of a double helix of the genome. So I think the it's particularly called as a CO1 gene. It's basically CO is for cytochrome oxidase 1 subunit, which is known to be a gene which is more stable. You see, I mean, to establish phylogenetic interrelationship. For example, what you see on your screen is just one single family of fish, and the multiple genera and the species affiliated to them. What they found very interestingly was, after harvesting and amplifying this particular gene, which was the cytochrome oxidase 1 subunit, this gene was found to be extremely stable. I mean, this is very interesting. It almost tantamounts to your barcode. I mean, if many of you know what is molecular taxonomy, where they amplify the entire genetic material and try to compare certain genes or certain fragments of the DNA. So if you across the board in this particular family of fishes, you find that this CO1 subunit gene seems to be constant, which indicates that there is a phylogenetic interrelationship. So that's basically what I'm trying to tell you. If somebody wants to know why this particular gene, or why those 3 genes that I spoke to you about previously, simply because the mutation rate is often so fast enough that you can easily distinguish between closely related species. I'm just trying to explain to you that this is only one gene which remains constant. You find lots of similarities. I hope this is very clear to you, that the sequence after amplification seems to be similar among conspecifics. Conspecifics essentially means individuals of the same species. Right. So this is basically, again, I mean, let's not kind of get too much trapped into the molecular basis of the whole thing. It's enough to know that molecular phylogeny or DNA barcoding today is one of the most important approach in. I think this is what I was trying to explain to you. eDNA, not many people talk about this. This is a very interesting and novel concept. Again, it's not at all practiced in this part of the world, but in UK, I mean, Europe and the US, I think this is one of the most pivotal and essential way of harvesting the DNA from environment that has been shared by animals, either from the feces or the hair or the dead remains or whatever, right? I mean, you know, when animals move, there is a lot of DNA that is transferred to the environment. If there's an abrasion, if there's bleeding, if there's some sort of a mechanical injury, right? So a lot of DNA from that animal, specific to that animal, is out there in the environment. If you know how to harvest it, and then, I mean, to collect it in the first place, then sort of how would I say, filter it and try to harvest it, then extract that DNA, after which you need to amplify that DNA, subsequent to which you sequence it. And once you sequence it, then you have a referral bioinformatic database of different, nucleotide sequences in the DNA of different species. So it's a program. All that you need to do is just feed it, and it tells you with which species does that sequence match. So look at this. This is very fascinating. You don't even get to see the animal, but by being able to sample, filter, extract, amplify, and sequence that DNA taken from an environmental source, you are able to construct something in retrospection that probably this animal has been here at some point of time. And mind you, this is also for animals which are probably no longer existing in that area for whatever reasons, right? So this is very, very, I think, critical area of technology that aids in retrospecting wildlife by sampling soil, water, snow, what have you, even air. You can access this environmental DNA because it's like a signature. It's the blueprint of a life which no longer probably exists there. At that moment, it is not there. It has probably gone away, but you can retrieve and retrospect. That's very interesting because a single sample containing environment DNA is enough to probably, I would say, comment on some kind of an endangered species status, whether it existed there at some point of time, whether it still exists. For example, the controversy about the pink-headed duck was going on for a long time. For a long we're talking about climate change, which is steady, progressive, right? We're feeling that. So how is that going to really impact these animals? You could also probably find out from sampling the environment DNA. It also alerts you to the impending threats. For example, new pathogens. You've got novel pathogens emerging. We don't know. We've never heard of this before. SARS-CoV-2, we never had that before. Look at it today. You had Delta, you had the Omicron, you've got new variants coming up. And all virologists say this is not going to end here. It's going to be with us for another 3 years at least. They say 2025 is when probably the whole thing may settle. So, this is very worrisome. Not just about our species, but the impact that this kind of climate change and novel pathogens can have even on the wildlife is something that demands your attention. It's very important. Overall health of the aquatic and the terrestrial ecosystems can also be understood from this. Well, this is a premise which I personally relate to in a big way. I have been into scanning electron microscopy as an important tool for taxonomy, using micromorphology as criteria for classifying animals. In fact, the images on the right are a part of my work to establish with the first definitive record of a whip scorpion, which I put on the right here. Again, what I spoke to you earlier was a whip spider. What I'm showing you now is a whip scorpion. Whip scorpion or vinegaroons, as you call them commonly, are again very archaic arachnids which have cryptic habits. They will not normally be seen out in the open. They are averse to light. They are crepuscular, and they secrete something which smells very pungent and aromatic. And currently I have a DBT-sponsored project on chemical ecology of these. You call them Europygi or tailed whip scorpions. Now, what am I trying to tell you? Electron microscopy, where you are able to resolve the functional morphology. What you see on the left is a micromorphology on the antennaiform legs, which are called the whips of a whip spider. Of course, this is not an Indian species, but if everything goes well, I think the first report of paper on similar lines on sensory structures on the antennaiform legs of Indian species of whip spider should be out in another 2 months' time, if everything goes well, that is. So, but the point here is, if you look at this, what cannot be caught by your eyes, because they are too small to resolve, the functional morphology. Based on these kind of structures, if you look at these whips, the so-called antennae-form legs, which are the non-ambulatory first pair of walking legs, really speaking, but they're no longer the ambulatory legs, but instead they have been endowed with and they have been invested with a lot of sensory equipment, which have been given certain putative sensory perceptions varying from, let's say, chemosensory, mechanosensory, and so on and so forth, right? Kinetoaesthetics, which is their ability to sense change in the body movements. So, I mean, that's fascinating. And that's wildlife as well. I mean, if somebody again says, why are you talking about whip scorpions and whip spiders? Is that wildlife? Of course, yes. Of course, yes. And there's a huge scope for bioprospecting in this smaller, the lesser known wildlife. So if you have an access to this kind of state-of-the-art imaging techniques, whereby you are able to there's a process involved. It's not that you just take it and put it on a slide. You have to do sputter coating. You have to coat the entire, there's a critical drying. There's a sputter coating with precious element like maybe gold or platinum. Is prohibitively expensive. But at the end of the day, what you see is an image like this, which tells you with great certainty what exactly is a particular structure meant for. As I said, a putative functional morphology. What is this structure likely to be aiding in is something that you would be able to establish only by using this kind of technique. So yes, microscopy and advanced microscopy like scanning electron microscopy. I think in this series we've had Dr. Sharda Sawant, somebody who's recognized for expertise in transmission electron microscopy, already spoken to us about oral cancers. So this is about scanning electron microscopy is again, it doesn't delve into the cell, it is only scanning the surface. So the surface structure, the topology, the microsculpture, the designs are more easily known by this. So this is again very interesting. Well, again, here is something that we have tried at our end, and again, keeping my fingers crossed, we are in the process of establishing some sort of a research facility for bioacoustics, particularly for bat surveys. And what you see again here is a small equipment which is now used increasingly by chiropterologists who are bat biologists. These are bat detectors. Very complex to something which is very simple and handheld. Just to impress, I'm not getting into the complexity of how do you resolve a certain vocalization. You need to emit signals which will match the frequency of the ultrasonic sound waves, and then that's how you know the bat is around. Sometimes it's very good because if you don't see the bat, but you are able to recognize its ultrasonic acoustic signature, because you are able to record it and amplify and change that into an oscillogram and a spectrogram on your screen, because you need certain softwares you can say with great, I would say, guarantee, and you can confirm that, well, it is this species. Of course, you'll agree with me. The pandemic times has brought in a lot of focus again on bat biology. So that's very important. To the right, you see a small image on the frog bioacoustics, which is what I had done about 5 to 7 years ago with 10 species of frogs in Goa. And bioacoustic signature is a very interesting concept in which you record the frog calls and you, I would say, analyze the entire vocalization using a certain software, which is what we had then and we still have. And you realize in terms of amplitude, in terms of frequency of both the oscillogram and the spectrogram, each species is something very unique, sonic character. So this is fascinating because once again, you don't need to really go to the field. All that you need is a good, reasonably good sample of, an acoustic sample or the croaks. You should be able to record that. And even in a cacophony or a mix of frog calling, you are able to separate, you can remove the interference. There are ways of filtering that entire acoustic sample. So just to tell you, bioacoustics again is something very, very fascinating. Biologgers, very interesting again. This is all got to do with miniaturized technology. and advancement of battery life, what you do is you actually have a small logger. By logger, I mean something which will constantly record. What does it record? It probably records the GPS, it records the movement, it records the trajectory of the flight, it records the climatic changes, it records even the cardiodynamics of the animal onto which it has been fit. I mean, that's fantastic. If there is a progression in a particular direction, acceleration, a kind of a continuous acceleration in a certain direction also indicates flight. So, a biologger is a compact device which you can fix on the, it's very tiny, it's miniaturized, it's not heavy. It's not something which is going to interfere with the movement of the animal. And if it does, then it is useless. Anything that you attach, like for instance, the lower slide, you see it's being attached to the fin at the base of that fin, right, of a marine mammal. So a biologger should be tiny enough, miniaturized, with all that it needs to do is keep on recording the various dimensions of interest to the researcher, which could be something from the environment or something insidious to the individual itself. I mean, there are some biologgers which can even record the changes in the hormonal titers. So, more or less, if there is a rise in testosterone titers at a certain point of time, and that's you link with the aggressive behavior of that particular, the males of that species, very clearly that the underlying testosterone titers are manifesting into that aggressive behavior. So what I'm trying to explain is that these biologgers is something mind-boggling. It's magical. These are archival tags where you don't they don't send you the information on a regular basis. You need to retrieve it and then decode that information by plugging into your, I would say, computers. So what does it tell you? As I said, and I'm just going to repeat that quickly, GPS location, condition of the environment is going to tell you the animal itself. I mean, it's cardiodynamic, it's like a smartwatch. Nowadays, everybody's into smartwatch. It tells you your SpO2, it tells you your cardiac rhythm, it tells you, a lot of the body temperature and so on and so forth. So this kind of a biologger is a wonderful aid, I think, for students of wildlife biology who would be interested in understanding. In real time, it records, but does not relay. That's the difference between, I would say, something like a radio telemetry collar, which I'm going to talk to you about in 2 minutes from now, and a biologger. So once you recover it, you are going to decode that information because you're going to transfer it. You're going to basically download all that data. So just a few examples, which I thought I'm just going to give it to you. Biologging is something fantastic today. That's the seagull, which has got a way, you can very clearly see on the back between the two wingspan. I mean, it's got a biologger, and what it does is it's got a satellite telemetry system, which can track the movement of the albatross by using this kind of it's like a beacon light. You've seen a beacon light on the ministerial cards. I think now that's not being allowed. It's something like that. It's a miniaturized beacon, and it constantly records. There's also this accelerometry, which is all got to do with the movement in all three dimensions. I mean, it's got vertical, horizontal, and whatever, altitudinal changes, absolutely everything. The three-dimensional movement of birds, the speed, the directions, whether that's correlating with some sort of I would say, cardiodynamic change that the animal is experiencing or the bird is experiencing. There are some amazing studies in the West where they've been tracking these movements, the pan-global movements. For instance, most of these migratory birds, they go through something called Zugdisposition. I mean, that's a German word, which means pre-migratory fattening. They take in a lot of food reserves. And there are some of these, like the Arctic tern, which never comes down. It's constantly flying at great heights, and there is no stopover site. So if there is no stopover site, how does it endure that kind of a long journey, really speaking? I mean, how does the heart really respond to that tremendous task of pumping blood for that long a time without any rest? Or if you've got a very smart biologger, then of course, even that can be intelligent miniaturized devices, basically, which allow measurement not only of the physiology, but also the oceanographic parameters, even in case of the marine mammals. Sometimes you record it and send it, or it can connect with the satellite. It's got a satellite transponder, which can relay signals. That's also possible. That's a combination. Then it becomes telemetry with biologging. But that's again very, very interesting. It gives you some understanding of what the animal is going through on account of climate change and change in habitat. This again very I'm so sorry, I think this got shifted, but doesn't matter really. Well, these are some of the candidates that we have, and I think it's all right, it just got shifted. But yes, for radio telemetry, which is again remote monitoring of animal movements. I've already spoken to you about carnivore movements. I'm not going to tell you much. I mean, this is the leopard, the dhole, the jungle cat. You've got a jackal here, and you've got the melanic panther, and you've got snow leopards. All these have been, in India, they've all been radio collared to understand their movements, to understand, I would say, the daily activity, budget of patterns, and so on and so forth. So basically, what exactly is that radio collar? I think I have to share with you somewhere around 2014, when I was in France, we had a hands-on module on use of these radio collars for carnivores. And that was pretty interesting. By Society for Conservation Biology in the United States had conducted a program. And trust me, it's very fascinating. Of course, there's some criticism that in India, especially, a few radio-collared tigers were eventually found to be dead. Accidents can happen, but that does not write off the radio collars as very important what should I say, equipment for monitoring animal movements. Nothing but a small, rather a wide band of a machine fit to a belt. You can normally put it on the neck, and it kind of emits. It's got a transmitter which is fit into that collar, which keeps on emitting signals at a certain frequency, which you can catch with the receiver. You can see the worker on top is receiving signals. So when the signals match with the frequency emitted by the radio collar of a particular tiger, and mind you, the frequencies differ, and that's how they differentiate. The frequency is set, and the frequency at which the signal would be emitted, and whatever amplitude and the other acoustic characters of the signal emitted by a transmitter of, let's say, animal X will not be the same as that of animal Y. So you matching those frequencies, because you got the referral database, you know exactly in which direction that animal is moving. It will start beeping. When the frequency matches, it starts beeping. And that's how you can track the animal. So this is like, I would say, you get a real-time understanding and assessment of the whole movement. That's very important. Very highly scientific and of huge conservation value, particularly in resolving human wildlife crime, in locating the animals. You see, the carnivores have a huge range. I think it's in several hundreds of kilometers, square kilometers. So to keep a track of an animal which is moving across such a large area, you certainly need to do it remotely, and that's possible. But of course, there is a small prerequisite, and which is to fit the collar. Certainly, you need to first capture the animal by using certain tranquilizing drugs, which you will shoot through the dart gun. And sometimes very unusual kind of experiences would be encountered by people. Either the dosage is inadequate and the animal could suddenly wake up, causing a flutter in the hearts of all the workers. That's an emergency. Or sometimes because of overdosing of the tranquilizing drug, the animal does not wake up. I mean, therefore, normally, once your radio collaring is done, whether the animal wakes up or not, you need to immediately inject an antidote. So that's again very important. The minute you pump in an antidote, it recover from the tranquilization effect and start, it's back to normal. It takes some time for it to gain its normalcy, but that's how it works. And the beauty of the collar is you don't need to do anything once you kind of put it in place for as many as sometimes 3 years, and as long as the battery lasts. You've got a lot of high endurance batteries now. The radio collar animal can be followed for a long time. Sometimes, of course, the animal can chew it off, or the conspecifics can chew it off for the other animal, or sometimes they just fall off. That's possible. You need to retrieve that. I mean, if you find that you're getting the beep and the signals constantly from one place, and the animal does not seem, the source doesn't seem to be mobile, there are two possibilities. One, that it has fallen off. Two, that the animal is dead. So sometimes this is very interesting. Even, the recent death of T12, right? The collar Wali. I mean, everybody knows collar Wali, right? In fact, she was called collar Wali because she had a collar installed. And I guess that is basically how they realized that it wasn't moving any further. There are some videos being circulated on WhatsApp, the last moments of collar Wali. Please have a look if you are able to. How did they know where exactly is collar Wali? Obviously by tracking her. Of course, she had a favorite destinations and locations. They are very site fiddle. I mean, that's again very important. They are territorial and demonstrate a very high degree of site fidelity. And she had a favorite place close to a pond where she would rest. And that's basically, but generally speaking, collar Wali was so called because she had a collar that allowed people to track her every single movement in the forest. So this is again very interesting. I just want to tell you this again. I mean, these are images that I've picked up from stories in my own state. And this is that Dandeli Anchil landscape, which was earlier called as the Kali Tiger Reserve. And I was just asking on the very first day a question to Mr. Vaingankar, member of the Wildlife Board in Maharashtra. And he spoke about the Tilari tigers. People in Anchi Dandeli and Kali Reserve say they are the tigers of Karnataka. People in Maharashtra say they are the tigers of Maharashtra. We are in between. We have to be happy and content by saying these are vagrant, I would say, tigers that just move between Karnataka and Maharashtra. So the question is, whose tigers are there anyway? Logically and reasonably speaking, no particular state can claim absolute proprietorship over any animal. It's a national asset. But the interesting part is if you radio color, you are able to know exactly where the animal is starting from and where is it reaching the ultimate destination. Where is it kind of going to den and where is it going to probably breed? I mean, that's something very fascinating. And more often than not, if you notice in Goa, all our protected area network is contiguous with the Western Ghats. So that's again very interesting in terms of using this radio collaring techniques and so on and so forth. The lower two images, of course, are the unfortunate revenge killings which happened around 2 or 3 years ago. If I think around 220 or end of 219, I remember. Among the pastoral communities, it was a revenge killing because the tigress probably predated on the buffalo, and that was the only source of income, I suppose, for the family, which was forest dwelling. And in a sort of vengeance mindset or for a vendetta, they left that carcass and probably topped with some sort of they say, sort of de-taking powder, and there are a number of hypotheses yet to become. I think the matter of sub judice would not be appropriate to comment on that. But the point is, revenge killings are realistic; they happen, and sometimes for the need to track the animal on a regular basis for ensuring its safety as well, you need a radio collar. So this is again very important. Radio collar would ensure that the animal will be contained. Anti-poaching technology. I think this is the serious issue that we all face in all of the world's wildlife-rich countries. And India is one of the B17 countries, which is to say it is one among the 17 countries with exceptional biodiversity and wildlife reaches. And therefore, we also invite a lot of, responsibility on our shoulders in terms of conserving what we have within our land. And if you look at the total landscape area of wilderness in India, let's just talk about India, or maybe for instance, in Africa and India, if you look at this, then the landscape is so vast that it is physically impossible for a handful of, the forest brigade to actively monitor and conduct a surveillance of animals on such a huge and a vast landscape. So what do you do, really speaking? You definitely need to, in a bid to dissuade the criminal elements from poaching the rhinos, for instance. I mean, you'll find that very curious that I put an image here, which is quite funny because you find a rhino with, I think if I am not able to see it very properly, some sort of a pink or a violet kind of dye has been used and the horn has been painted. The idea is very simple. This is a dye which has been injected in the horn. And this is a dye which will be detected at airports, even if you try to smuggle the rhino horn by powdering it. I mean, the wildlife criminals come up with very ingenious ways of smuggling things. They will not carry it out as a horn. They will powder it because ultimately the rhino horn is believed to be an aphrodisiac, and it's very popular in Oriental medicine. It is not to be necessary to be taken out as a horn itself. You chop it, or you chip it, or you powder it. But if it is dyed, they had this experiment done whereby a certain element was, certain dye was injected in the rhino horns just to dissuade the criminals from smuggling this out. So even if they kill it, I mean, this is not preemptive, really speaking, but it only prevents trans-border smuggling of this precious rhino horn. But there is something more interesting now, which I have shown in a small sort of an animated kind of figure there. This is something really, really, I mean, interesting, and I think that's the crux of the whole discussion on use of technology in modern research of wildlife biology. What they are doing here is they fit a miniaturized device. It's a combination of a GPS locator. It's got an alarm. It's got sensors. It's a kind of a biologger. It's all in one, which is implanted in they drill a small hole in the rhino's horn and they fix it there, and that's it. So for whatever reasons, if the animal is poached, obviously what will happen is with panic reaction, the minute the animal is being chased or is even being chased, forget being shot, then the minute starts galloping or starts running fast, there's going to be accelerated heartbeat, which will be sensed by the sensors in the biologger, which sends through a satellite transponder a message to the satellite, which will immediately send and relay that message forward to the monitoring station, right? I mean, where people are constantly logged in and monitoring in real time the movement of that rhino. The minute they see that the heart rate has shot up in this particular area, this particular individual for which they have a code and all the parameters are being monitored, they convey, they relay the message further to the SCOD, to the mobile SCOD, and it's out there in the African continent. Is you don't access that site by on ground, but rather they've got the choppers, they've got helicopters. So in a matter of minutes, I would say the chopper would reach that place and take the necessary action to rescue the animal from the poachers, or if the poaching is accomplished, at least the poachers would be arrested and prosecuted. So this is amazing. And mind you, this is not just an imagination. This is actually happening. This is already happening in Africa. It is yet to come to India, though. But I think that's in the offing now for the rhinos in the northeast, Kaziranga. So this is brilliant again, that you sit in your field station or wherever, or at your headquarters, but you're logged in, and you are connected to the satellite through the transponder that's sending messages continuously from the biologger kind of. It's a compact all-in-one gadget, which has got a sensor to, even if the animal is speeding, accelerometer. I mean, if it's why is it running really speed? The panic triggered cardiodynamic alteration. Everything is sensed. Sends it to the satellite. Satellite will relate. It will further relate to the mobile SCOD for a quick action. Brilliant, absolutely. That's basically the anti-poaching technology. Let's talk further about, is it happening in India? Well, yes, in terms of UAV, which is unmanned aerial vehicles. You do have a lot of research happening at Wildlife Institute of India. They certainly are using a lot of these unmanned aerial vehicles, so-called drones. There are some restrictions, of course, on use of drones. That's become in great fashion today to use drones. I mean, I remember my times used to fly kites. Nowadays, kids fly drones, they say. Which is very true, because drone technology is more easily affordable now and easily available too. But now the government of India has come up with a lot of restrictions on not allowing usage of drones in certain sensitive areas where you may have defense installations and so on and so forth. So, I mean, this is very interesting. What is the use of a drone? A drone does what you and I would not have been able to do, give you an aerial view. It's like having an eye in the sky. It's like having an eye in the sky. So you go up, the minute you raise yourself at a high elevation, you got a wider field of vision. And it's relaying information directly even into your mobile. So I think the enforcement agencies are at absolute ease. They know exactly what's happening. They're getting a real-time data being released by the drone. It can capture, it can relay it, all kinds of things. So, I mean, in areas or in systems which are constrained by tight budgets and fewer field personnel, next best option would be using unmanned aerial vehicles or drones, which will quickly, sort of bring under visual surveillance areas which could have otherwise not been monitored by oversight. So this is again very. These are images all from the Wildlife Institute of India. Now I want to tell you a very interesting story. What you see here is a crested tern in the bird on the left is a crested tern in Adelaide Islands. And you got this man, Gerard Hatson here on the right, who's doing something, right? On the lower image, you see he's laying something on the seashore. What he's laying on the seashore are nothing but decoys or rubber ducks. It's a very interesting experiment done by the scientists to confirm, or rather to establish, that surveillance by unmanned aerial vehicles for the purpose of enumerating population and for monitoring is more confident than that done even by human eyes. Absolutely, I don't know how to look at it, but this is fantastic. He proved in a brilliant paper again in 2020 that what the drones can do in terms of accuracy and confidence is almost to the tune of 96% accuracy as compared to trained bird watchers. If you got trained bird watchers, ask them to enumerate a particular area of birds and do the same thing using an UAV, he says there are more errors done by experienced birders than by the drone. How did he do that? He used this kind of rubber decoys to create a sort of, a predetermined matrix was already there on the beach. In the same way, he laid down all this and he flew a unmanned vehicle or a drone, and he compared the results of the two teams. The team on top are all experienced birders. They've got their field scopes and everything. And then I didn't put the third. It's astonishing that the artificial intelligence sometimes can overtake your human cognitive abilities. That's scary, but that's fascinating as well. So I think this is again just a confirmation. Are you able to hear me? Because I'm getting a message, my internet is not stable. Are you able to hear me, please? Dr. Gaikwad, are you able to hear me? Anybody?
Yes, sir. Yes, sir.
Thank you. Recently in my system, we had this international webinar on marine mammals and their conservation concerns. My colleague, Dr. Preeti Pereira, was the convener of that. And that is when we got in touch and discussions with some of the topmost marine mammal experts from all over the world, in fact, including US. Something which they often do in terms of invasive sampling is, you see, one of the most difficult aspect of sampling marine mammals is that you need to collect either the tissue sample or blood sample, which becomes very invasive. You need to, sort of shoot them with some sort of equipment to retrieve that for tissue samples. Now, with this drone technology, you can go in for remote non-invasive sampling. Absolute dream come true. What do you see here is very interesting. What do you see here? The biologist Lane Caro, Ocean Alliance, was the one who pioneered the first technique ever, which he very fondly called as a snot bot. Snot is all the nasal, the expulsion. You see, they have a blowhole, right? The marine mammals, so which, I mean, the whales in particular, through which they periodically, sort of sprinkle and shoot a lot of water up in the air. Now, they need to breathe, they need to surface, obviously. So what this gentleman, who basically was a trained biologist, did by handholding with the remote technology people is, he said, let's design a drone, which was more of a, you call it a quadcopter. A quadcopter would mean it has got a set of 4 propellers, which you can very clearly see in this image. And it is fit with a Petri plate at the base. All that it has to do is fly over exactly over the whale when it is snotting it out, you see. When the whale is blowing the water into the air, the drone is just positioned exactly to pick up that sample, because there is a Petri plate and the requisite holding equipment. So it will fly through the whale blow and pick up the samples. Obviously, since it's coming from the blow hole, what do you expect to be contained in that sample? It will have a lot of biological material, including DNA. It will have, that's very interesting, microbiomes, the kind of microbes that reside in the body of the whale would also appear in that sample, in addition to which even hormones. I mean, fascinating that the don't need to take tissue samples at sea using invasive process. I think this is something which really kind of impressed me the most in terms of remote non-invasive sampling using drone technology. So I thought I would share that with you, of course. These are, of course, intermittently, I'm showing you a few images of the kind of biodiversity reaches that we hold in our forests again. This is just to show you. Well, now, there are a few interesting contemporary challenges too. For instance, there are a few research laboratories in the world which is now looking at the impact of climate change, the impact of habitat alteration, the impact of environmental contaminants on the health of the wildlife on the left. So what do you need to do to be able to ascertain that reproductive events are happening properly, to be able to ascertain that the populations are really surviving, to be able to understand it by recalcitrant molecules of chemicals which are entering their bodies through a food chain transfer, right? I mean, if you recall the classic book of the 70s by Rachel Carson, The Silent Spring. The Silent Spring is a nightmare even in 2022, though the book was written in the 70s. A lot of these engineered molecules which are recalcitrant, which means they're not easily degradable, environment, they eventually get picked up and they start getting assimilated through the metabolic events. And this, I think it's going to cause a metabolic arrest and a lot of metabolic disruption in the bodies of a lot of animals like birds and amphibians. So that's again a contemporary challenge, and which is where you need a lot of physiologists. I want to say this, Shivaji University has is in legacy of Professor Varute, a very renowned, I presume physiology was one of his forte. So yes, I mean, if somebody says, what do you do with physiology in wildlife? I think that's one of the key essence of any wildlife investigation today, given the fact that physiology tends to be disrupted by all kinds of anthropogenic changes, whether it's climate change driven by human activities, whether it's got to do with survival question marks, it's got to do with altered distributions in terms of animals. Now, this, what you see on the extreme right is something interesting. A snake species, which is now in an altogether a new environment, a different kind of a climate regime, which you would never expect that snake to be found in, but probably that is the only climate refugia, which means it's now adapting to new kind of climatic combinations. It has no other go but to sort of acclimatize. Right. So this is again something very fascinating. So I just thought I would share that with you, that physiologists have a major role to play with all kinds of state-of-the-art equipment in physiology, whether it's electrophysiology, whatever. Right. I think that's again going to complement our understanding. I just thought I would say you this, show you this, that we have used geographic information system in writing our management plan in Goa as well. I mean, this is about the terrain features and the contours. This is about land use patterns. This is about creating a fire vulnerability map, and this was about a management. Not much here, of course, but I think this is very interesting. And this is one of the one of the things that I often speak about in my classes in regular curriculum is the concept of frozen zoo. But I've never explained that with any sort of graphics ever in any of the conferences. So I thought today I should explain that to you. And well, Frozen Zoo is an initiative of San Diego Alliance. I mean, there is a group of people there who specialize in different domains. And a frozen zoo is not meant to be, as is often believed, a zoo which is at high altitudes where the climate is very frigid. That's really not what it is. I mean, it's maybe a zoo in Himachal Pradesh is not eligible to be known as a frozen zoo because the climate is frigid. A frozen zoo is nothing but an ex situ initiative in which the germplasm of critically endangered, or even for that matter, extinct animals is cryopreserved. What you see here on the, I think, I am so sorry. I think that's going to come next. I'm just sorry. That's going to come next. Well, I think this is about ultra-modern zoo. And this example is from a zoo that I have visited in Japan. It's one of the best zoos in the world, called the Ueno Zoo in Tokyo. I spent nearly around 2-3 days at this zoo studying the way they function and the way they work. I've documented a lot of their very interesting innovative. One thing which impressed me the most in this zoo is their facility for assisted reproductive technology. I think Dr. K.C. Avari spoke about ART in human beings, but mind you, infertility is something which afflicts also the animals. And very recently, I think one of the giant pandas delivered twin cubs, and they were prematurely delivered and needed a lot of artificial support, including incubation and so on and so forth. And the image on the left is again an ape, which is taken great care of in practically a neonatal unit and a state-of-the-art equipment there in terms of thermal incubators and what. And look at this sense of commitment of the staff who's holding that ape young one, which is probably wired to a lot of things. I think that kind of a commitment, of course, demands in the first place great passion, a great deal of expertise, and of course, a lot of funding, which is a little difficult to dream in a typical Indian situation. And I'm sorry about that again, because we all identify with this place. We have our own problems. And I'm sure you know that in our country, this kind of ultra-modern facilities for prematurely born neonates of our own species also is something that only a few people would be able to afford and have an access to. But today, in many parts of the world, I mean, in the developed countries, these facilities are also extended and established in the zoos. We all accept that a zoo is one of the finest ex situ conservation initiative, not only in terms of educating people, but also conserving species which are probably on the, I would say, verge of extinction or have even gone extinct. For instance, in Mysore Zoo, we have a cheetah specimen. It's an African cheetah, but we do have a specimen, which otherwise is not there anywhere in the country, maybe only in zoos. So just to impress upon you the importance of this kind of ultra-modern zoos. And now this is what I was trying to talk about, the frozen zoo. The frozen zoo has nothing to do with the zoo. A frozen zoo essentially is the largest and the most diverse collection of germplasm, the first of its kind in the world, and that's in San Diego. This is very interesting. What do they have? I mean, if you see the entire team on the top left, they are just trying to open up a sort of a cryo unit, which has got the germplasm stored, as you can see beneath on the lower slide. This is holding something like 10,000 samples of living cell cultures, oocytes, sperms, embryos which are frozen, representing nearly 1,000 taxa. Can you believe that? That somewhere in the world, we have this kind of an exceptionally rich, it's what you conventionally in our, but this is more particular. It's putting an elephant and a tiger and an equine species in ice cubes. Basically, what I'm trying to suggest is only that if you are uncertain about the conservation future of a certain species, the least that you can do is spare the germplasm of that species to this particular consortium or oocyte with a cryopreserved sperm, whatever. This is basically in the domain of assisted reproductive technology, but it has a huge implication in understanding evolutionary biology and wildlife medicine. So I thought this would be very interesting. I am not sure if I've got anything. Well, I think it's exactly 1 o'clock, and I think you had given me 2 hours, Dr. Gaikwad. I started about 11, and it's showing me 1 o'clock in my watch. So I think
exactly 2 hours.
I'm not trying to suggest that I have an answer to everything. I may not have an answer, but that should not prevent you from asking a question. Thank you, and I really appreciate this opportunity. Very kind of you.
Thank you very much, sir. Thank you.
Yes, sir. Pleasure always.
Yeah, we'll take a couple of questions here.
By
If
all
anybody
means.
is having questions, please ask.
I hope I have not violated your time limit. You gave me 2
No,
hours
sir.
and I spoke for 2 hours.
No, sir. No, sir. Initially, you said that 2 hours, but I know that your potential, you can continue till the evening also.
Yes, now I can see you. I would be really happy even if there are any observations or submissions. And I don't expect everybody to agree with my views. You feel free to disagree with me also, that's fine, absolutely. But I sincerely hope I was able to, put before you some new ideas today and emphasize how technology could complement our efforts at saving some of the world's most endangered species, whose conservation future is very precarious.
Yeah. Now participants can unmute themselves. So by unmuting yourself, you can ask the questions.
Yes, anybody. I see a good number. I think you please check your chat box.
Yeah, I am checking, sir.
If there are any questions, then please.
Yeah, there is one question from Varshat Chaugle.
Yeah, I was reading the same thing, whether radiation effect on animals or radiation mutation as a carrier. Madam, these are not ionizing radiations. The signals are radio signals. It is the same thing by which we receive our FM, stereo, and whatever. So please don't get me wrong. When I speak about emitting signals, I'm not saying they emit radiations per se. I hope that answers Dr. Varsha Chaugle. I mean, she's a student probably, Varsha Chaugle. Does that answer a question? I'm not sure. See, I mean, I take it a little further. As a matter of fact, a certain amount of background radiation is what we're always exposed to, and that is necessary to ensure genetic fluidity, right? Because you wouldn't have variations without, or a genetic flux without being exposed to that. Optimal amount of background radiation is always very important, but it's only when the rays are ionizing radiations, start knocking out atoms from electrons and make the entire matter very unstable and reactive, then you have this, or knock out the whatever, the DNA molecule, then you could have mutations. But insofar as the biologger or the transmitters that I'm talking about in radio telemetry, don't go by the word radio and radiation is not one and the same. I hope that is. Sir, Azun, any other?
Sir, good afternoon, sir.
Who's this? Please identify yourself.
Sir, my name is Nagesh Birazar. I'm a research scholar.
Yes, Mr. Birazar, please ask.
Sir, I have one question that sophisticated technology needs funding.
Absolutely.
And funding empowers work. So
funding,
what
I'm sorry, I didn't get it. Funding empowers
work. So what are the ways and methods to pour in some good funding to work on the subjects of interest with perspective to wildlife conservation?
Mr. Birajdar, I am extremely sad and sorry to tell you that I have been the biggest victim of fund deprivation as such, but that has not stopped me from collaborating with people. I think the best way to tide over this fund crisis is to handhold with people who have got funds. Today, the key to progress today is not to work in isolation, but rather to forge partnerships. Let me say that very clearly, and trust me, if you are passionate and if you are genuine, people will always want to extend that support to you. Trust me, believe me, till date, though I've been using electron microscopy extensively, I would say, scanning electron microscopy. And I'm very fortunate because we've had this facility, of course, in Goa in 2 institutions, wherein there is a charge and whatever. But if you are a committed academician, I think with their discretion, they waive it off. They waive it off. And even a lot of these workers abroad, I'll just give you one example. 3 days back, I was doing my literature survey on something, right? And there was a paper which has been published only last week. On the website says you will pay $36 US dollars to download that PDF. Are you hearing me?
Yes, sir.
So that is anywhere around 2,500 rupees. So I sent him a very polite mail, dear so-and-so, I understand that this paper is not freely available and I need to download this, but that costs me around this much and I'm not able to afford that. No pretense, I'm not able to afford that. Could you please send me a PDF or a soft copy? Believe me, by evening I had the PDF with me in my inbox from all the way from Colombia. Not only that, if the person further asks me, can I be of any help to you? Could we collaborate?
That's wonderful.
Absolutely. So I think it's all got to do with your approach. If you take a high pedestal and start, talking as if I am the benefactor and you are the beneficiary, nobody wants that. Keep a low profile and approach people. I'm sure it is done. I hope that answers your question.
Thank you, sir. Thank you very much.
Okay, sir. There is one question in the chat box asking Jasmine Mujawar.
Yes, sir.
What precautions should we take when putting trap cameras in the field?
One is ideally it should be at an appropriate level that the animal would be trapped. In the sense, the image of the animal would have to be, I mean, you put it at a wrong height, at a place where it's very susceptible to being pilfered. I can tell you, many camera traps are stolen by people, by the way. So one is you need to try to put it in a place where it wouldn't be easily seen, but at the same time in a strategic place where the trajectory of the animal should reflect in the lens. That's very important. That's the most important criteria. Nowadays, you also get a lot of shelters for the cameras, additional reinforcement, so that they can endure the climatic changes.
Okay, sir. And one more question. Is there any impact of those?
Just a minute, Sarah, to answer Yogesh Mane, invariably when you decide your location for camera trapping, the wisdom of your beat guards is always taken into consideration. I mean, if it is within a protected area. If not, the locals or anyone who is reasonably familiar with that area, see, animals are normally habituated to take certain, I mean, they have an exploratory urge. For regular commuting, they invariably have a fixed pathway. So if you know by previous experience or some reliable sources tell you that this is where the animal generally, sometimes you can randomly also, rather you have to put some camera traps randomly, but ensure that the height at which it is installed, reinforcement, proper strapping, additional, I mean, nothing that would impede the lens, and the level is very important. Yes, sir. What is the next question?
Is there any impact of those devices on animals?
Well, yes. Of which devices? Camera traps.
Might be. Question from
I'm not
Pallavi
sure if it's talking about the radio collar. Well, yes, they find it a little bit of an encumbrance initially, but they get used to it. Even for that matter, leg bands. When we put leg bands in birds or sometimes even in other animals, it shouldn't be too heavy that it interferes with the movement of the animal. It shouldn't be too bright or flashy that the conspecifics refuse to let that tagged or banded animal in the group, right? It should not be made of any kind of toxic substance that there would be a sort of a cutaneous transfer. These are some basic apprehensions and care that we need to take.
Okay, sir. Dr. Mangal Chavan is asking, radio waves are a type
yes,
of...
absolutely right. Absolutely right. And I completely concur. In fact, absolutely right. She's right. But you need to adjust those frequencies to a level that it will not cause hyperthermia. In fact, hyperthermia has been reported in radio-collared animals. I completely agree with her. Absolutely right. Radio collar can put animals into, yes, sometimes they get asphyxiated. I think with one of the finest tiger experts of the country, Ullas Karant, also we have had accidents happening with tigers, and he has come under a lot of fire. So yes, I think these are professional hazards and errors that can happen anywhere and with anybody. So that is yes, the answer is yes.
Yes. Yes, one more question. How can we overcome the human-animal conflict? For example, tigers which are moved towards village are because of intra-specific struggle between their own species.
I think I will not give a specific answer. I would rather give a very generic response to this question. I think most of the human-wildlife conflict arises from conflict of resource sharing. It's basically a competition between the animal and the human being for whatever reason. Sometimes you find a tiger attacking when people enter the forest for collecting firewood, or maybe even for hunting or poaching. Or sometimes the carnivore may come out in search of reasonable, I would say, catch per unit effort, in the sense of the returns it gets after hunting a certain prey. Disturbed prey base, poor density of prey base, these are some of the issues. But I think the first thing that we need to do in terms of resolving human-wildlife conflict is, number one, try to minimize proximity to wilderness. We always, I mean, it's the same question, the chicken first or the egg first. I think over the last few decades that we have been, I would say, getting swept by this wave of urbanization, we are getting closer and closer and closer to wildlife areas. That's one of the major reasons as to why, the conflicts are on a rise. It's an interface, first of all. Human-wildlife conflict interface needs to be recognized, identified, and mapped. Only then you can intervene in terms of mitigation. I'll just share one simple experience that I have personally. There is something called non-lethal carnivore exclusion barricade, which means by altering the landscape, you can also keep the animals away. That is also possible. And some basic, I would say, precautions that need to be taken, let's say, for instance, by herders, animal herders. The impoundment has to be proper. Sometimes if you tie the animal outside, that will obviously be like a bait for the carnivore, right? So, I mean, I've seen this happening in Gujarat, or even in the northern part, in the Ladakh range, and the Spiti Valley, where there are some of these sheep herders, Rabbaris and all. I mean, a lot of those hill dwellers. I think the impoundment facilities are inadequate, and that is what brings in the snow leopards. And similarly, here in the peninsula India, it's the same story. I mean, even if you go to the arid zone, or Gujarat, Rajasthan, attacks by leopards, attack by lions, basically is all essentially because it's an easy prey. So if the carnivore gets an easy food, then why should it struggle and chase an animal? In Gujarat, it's a different story again. There is a lot of spillover, Dr. Gaikwad. I mean, the metapopulation habitat, the carrying capacity has been violated long back. So the prey base is not commensurate with the number of lions there. So definitely they will come out in search of prey. So it's a very complex issue. Human-wildlife conflict resolution is a challenge of all wildlife biologists, and there isn't any quick fix towards that. There is no one single reason why it happens, nor one single solution to it. It's a very complex issue. It's got to do with your culture. It's got to do with your terrain. It's got to do with your geography. It's got to do with the kind of wildlife you have in your place. So it's very difficult to, I would say, give a straight answer to this question. It's very complex.
Okay. Thank you, sir. Thank you very much. Thank you very much for a wonderful talk and excellent explanation.
Thank you, sir. Thank you.
With this, I invite Professor Bilave, sir, for further formalities. Thank you.
Good afternoon, everybody. I hope so you can listen to me. I'm here to express the vote of thanks. I take this opportunity on behalf of the organizers to express my vote of thanks to Dr. Manoj Bhavkar, sir,
Thank you, sir.
Manoj Borkar sir has very nicely synchronized the concept of the technology-driven wildlife research. He has started with the CT, that is the camera trapping, then headed towards the ACM, biologgers, bioacoustic, radio telemetry, and finally he has come up to the frozen zoo. Unless and until you have the strong field work, strong homework done at your place, the way Dr. Manoj Borkar has done, I don't think so the barcoding and the gene sequencing can work unless and until you know what is there in the environment. And as sir has very nicely synchronized the technology and the work done by him in the wildlife himself. So thank you one and all, and over to Dr. S.M. Gaikwad sir, Organizing Secretary, please.
Thank you once again, Borkar sir.
Pleasure, sir. Pleasure. Always a pleasure.
Thank you.
Thank you very
now,
much, sir.
Yeah, now you can leave, sir.
Sir, what time do we reassemble for the valedictory, if I may know?
Yeah,